All right, I think we will get started this morning. So I don't know, how many of you were here, I don't know whether it was 4 weeks ago when I did the first half of this? A few of you, not all of you. Okay, so just for those of you who weren't here, I gave a primer on sort of variant interpretation, and the intent was to do both variant interpretation and gene interpretation, and we never got to the gene part. So Chris kindly scheduled me again to come back and do this sort of gene half of this lecture. Although I did, now that I have a whole nother hour, I added more to it, so with a little more meat. So we're going to really talk about not the actual scientific discovery of genes and their role in disease, but once someone else has made a discovery, how do we evaluate the evidence and decide whether it's ready for clinical use? That's more of what I'm trying to focus on today, is really how we think about bringing gene discoveries into clinical implementation and evaluate their relevance. So if you haven't read this paper, it's a really nice summary that Jessica Chong and Mike Bamshad's lab wrote, looking at really the state of rare disease discovery and the genes and our understanding of their role in disease. And what you see from this is about 15% of genes to date have been identified to underlie Mendelian phenotypes. There's another 3% for which the conditions have been mapped to a locus, but the gene hasn't yet been identified. About 30% of genes are predicted to result in embryonic lethality, and so we wouldn't necessarily observe humans with mutations in those genes unless we look early on in development. But then about half the genes, we still have no idea what their impact leads to in terms of possible human phenotypes. And of course, these are all candidates for thinking about Mendelian disease. You can also see that the methods that have been used to implicate these genes in disease have transitioned from conventional approaches to now largely being done by exome and genome sequencing in the last few years. Now, as we think about the use of these genes in diagnosis and clinical testing, I took a slide from Daniel, modified it slightly, but we have variants in genes that we understand well. So this Delta 508 is one of the most commonly understood pathogenic mutations for cystic fibrosis. So it's in the variant level evidence is very high, but also the gene level evidence is very high. We understand the CFTR gene as the causal gene for cystic fibrosis. So it sits up here in the right corner. But then, as we talked about a few weeks ago in interpreting variants, there are many what we call variants of uncertain significance, or VUSs, in well-understood genes. So it doesn't matter that you understand the gene and its role in disease, you're still going to find variants all the time that you can't interpret. And so on this axis, these are for genes we do understand, but the variant level evidence ranges. But there's this other axis here, which is the gene level evidence. And so we can find plenty of variants of uncertain significance in genes of uncertain significance. As I just pointed out, most genes, we actually have no idea what they do with regard to a human disease phenotype or even biological function. And the most important thing here is that you cannot interpret the pathogenicity of a variant if you don't understand the gene. So if you don't have good evidence that a gene is causally implicated in a disease, you cannot call it pathogenic. You can say that the gene does not have a function because it's deleted. So you can look at variants that might presume to lead to a null, but you can't say it's pathogenic, because pathogenic implies for a disease. This is pathogenic for cystic fibrosis, or pathogenic for cardiomyopathy, or pathogenic for hearing loss. You cannot use the term pathogenic, even if it's an obvious null effect, if you don't understand the gene level evidence, okay? So I'm going to go through a particular case to kind of illustrate how we think about gene level evidence. Oh, you know what? There's, I think, an annoying advance thing going on here. Oops, that's not what I meant to do. This is clear timings. So how timings get on my slides. I don't understand. All right. Sorry, I will stop any of these slides going by themselves. I like to be in control. So this is just an exome case from one of our baby-seq studies. This is a real case, so a healthy newborn born in the Brigham Well Baby Nursery and no relevant family history. And we identified a heterozygous predicted loss of function variant. So this would predict a frame shift with subsequent termination fairly early on in the protein, and it's in a gene called VCL. So, whenever we find a variant like this, we ask a series of questions. Is this a disease gene? Is there valid evidence that this gene has a role in disease? If we check off yes for that, then we can go on to the second question, is it pathogenic? And then in this study, we go further from that, like, is this actionable? Should I actually return it to a baby? So if we start here at this level, this gene has been reported as a dilated cardiomyopathy gene. It's in OMIM. Doesn't mean there's good evidence. Someone has published evidence. So what is the evidence? Well, there's a zebrafish model. The VCL is expressed in the intercalated disc, sarcolemma, and Z disc. A transient knockdown using morpholinos demonstrated a sort of DCM-like phenotype in zebrafish. There's also mice with cardiac myocyte-specific inactivation of this gene that leads to sudden death. Half of the death before 3 months of age in mice. And mice with early sudden death, there were no signs of cardiomyopathy, but were found to have rhythm disturbances, suggesting VCL deficiency disrupts electrical conduction of the heart to changes in contractile dysfunction. Now, some mice survived longer periods and eventually developed a sort of a DCM-like phenotype. And there was one missense variant reported in a hypertrophic cardiomyopathy case, but no other reports related to hypertrophic cardiomyopathy. The VCL was actually initially implicated in dilated cardiomyopathy based on a single in-frame, single amino acid deletion in one patient, though later on the clinical significance was disputed when it was found in 0.5% of African chromosomes in the EXACT database. So subsequently, there were about 5 missense variants in VCL that have been reported in DCM patients, but very limited segregation data. So these are 3 of the pedigrees. Here's this in-frame deletion shown here, but this is that variant that was essentially ruled out due to reasonably frequent occurrence in African individuals. Here's another missense found in one proband, and then one other missense variant found here. It did segregate in both a child and a ant, but only 2 segregations there. And then it was also found in a large pedigree, although there was another mutation in MYBPC3 also segregating in this family, so it was a little unclear. So at the end of the day, the evidence that's present, there's some animal model data, but it's not a perfect match for a dilated cardiomyopathy phenotype, although you don't always assume animal models will exactly recapitulate human phenotypes. The human data, a bunch, like 5 or so missense variants found in cases is not strong evidence. We find missense variants in every gene in lots of people when we go sequence them. So showing that a single missense variant is truly causal is sometimes difficult without good de novo occurrence, segregation data, functional data, et cetera. So at this point, looking at this data, we would say, all right, there's actually limited evidence that this gene plays a role in disease. There are no loss of function mutations that have ever been described. And we would not go further with this and returning it to a patient based on the evidence that is collected to date. However, we subsequently, in my lab doing clinical testing, we had included this gene in a panel and we're testing patients with cardiomyopathy. And we came across what is now over 6 mutations shown here that are all loss of function. So 1, 2, 3, 4, 5, all 6. They're nonsense, frame shift, and canonical splice site mutations. We also have a series of segregations, 1, 2, 3, 4, 5, 6 different segregations that build further evidence for these variants. Now, not every patient with one of these loss of function variants is actually affected. You can see some individuals in these pedigrees, a parent who's unaffected here, a sibling and parent and grandparent who are unaffected. Although dilated cardiomyopathy is not a fully penetrant disease for almost any gene that's been identified. So this is not necessarily evidence against that. But everybody who has the mutation or has disease in these families does have the mutation so far. So, and this is just a list of all of the different loss of function mutations we've now identified. And the other thing that you often look for is consistency in phenotype. So all of the genes that are on our cardiomyopathy panel, we test in patients with a variety of different disorders: hypertrophic, dilated, arrhythmogenic, left ventricular non-compaction, restrictive, et cetera. And so usually when we find mutations that are not causative, they randomly occur in the different patients with different phenotypes. In contrast, when we find variants that are pathogenic, they almost always are found in a single phenotype. So what you see here is that pretty much all of these patients have dilated cardiomyopathy, and many of them have infant congenital onset, which is actually not as common in DCM. So now, with a whole lot more, particularly human evidence and better mutations, loss of function mutations, we've now been able to build stronger evidence. We would consider this now a gene with strong evidence for role in dilated cardiomyopathy. And then we can go on to the second question of, okay, is this particular mutation actually pathogenic? So in this case, the challenge, and we did go over this for those of you who were here a few weeks ago, in terms of interpreting pathogenicity, when you find particularly a novel loss of function variant, you can't just assume that it's disruptive to the protein. There are a series of questions you need to ask. The first is, are null variants an established mechanism of disease for that particular gene? And with the slide I just showed you and a whole series of loss of function mutations, we can say, check, yes, that's true. You can also now use the database launched here at the Broad by Daniel MacArthur's group in terms of looking at, is the gene constrained for loss of function variants? Because if they're pathogenic, then the gene should be constrained, particularly in a dominant haploinsufficiency state. And the answer is yes, the constraint score is 0.99, so quite constrained for loss of function variants. Now, this particular variant has not been previously reported in any human patients. It has been identified in 0.004% of exact, but based on the pedigrees I showed you with some reduced penetrance that you probably saw there, it wouldn't be surprising that an extremely low frequency but present number of cases could exist in the general population. We also, in order to say that a loss of function variant in a particular exon is going to be disruptive, we want to be confident that that exon isn't spliced out in some functional versions of the protein, meaning that it's not really that impactful. So we often look at all of the transcripts. Is this particular exon ever alternatively transcribed? And are there other pathogenic variants in the exon which implicates that specific exon in disease, in the biological function of protein? Now, because this is such a new gene and not well studied, there aren't actually any other pathogenic variants in that exon, but we can say that this particular exon does not appear to be alternatively spliced because there appears to be no described transcripts without this exon. It's also upstream enough from this C-terminus that we would predict nonsense-mediated decay would probably happen with this transcript based on where the stop codon would occur. And there are 4 predicted null variants that have been reported downstream of this particular site, such that we wouldn't necessarily expect a truncated protein that's this truncated to be okay. So we kind of go through all the checks for the novel loss of function question. We've checked them off, and at this point, if you go to the ACMG rules for variant classification, you would, with this information, lead to a likely pathogenic interpretation for this variant, which would then lead us to consider it for return to a patient. Okay, so now we're going to not really dwell on variant interpretation. As I said, today's lecture is really on gene-level evidence. And how do we use gene-level evidence? Why am I spending an hour talking to you about this? Well, one way we use this information is to guide inclusion in disease-targeted panels. When we put a test on the market to help diagnose patients, it's really not very useful to put genes that we don't understand on them if you're trying to return a useful piece of information to a patient. We usually try to not include the genes with insufficient evidence, include only these genes with sufficient evidence such that we could interpret at least some variants as pathogenic. So now you can look at this figure that a colleague of mine put together for, these are 14 different cardiomyopathy tests on the clinical market for diagnostics today. Some panels have up to 90 genes and others have less than 20. Why is that? Largely because there has been no standard for the level of evidence to put a gene on a test. And that is one of the challenges that we are trying to fix today, is really helping guide this process. And we've kind of come up with an approach, which is to say, if you're doing predictive tests like that newborn, healthy newborn that I'm about to give a result back to and say you're at risk for dilated cardiomyopathy, you better be pretty darn convinced that that patient is at risk. And so we would argue you should probably only include genes with strong or definitive evidence for a role in disease when you're doing predictive testing, telling patients they're at risk for something they don't know about. If you're doing diagnostic testing, that's a patient who has disease, has shown up in clinic and wants to know why they have disease and perhaps what treatment plan they might implement. I think it's fine to include these genes with moderate evidence that are pretty likely going to turn out to be strong roles in disease, and perhaps including them in panels, those cases will let you get over the borderline to strong and definitive. But we would really suggest restricting those genes with only limited or in fact no evidence to genomic studies where you know you're going into the unknown when you sequence a genome or exome. So that's sort of the concept that we have in terms of how these genes can be used in clinical diagnostics. And when you are doing exome and genome, by understanding the level of evidence for the gene, it helps you more rapidly interpret a given variant you would identify in the interpretation of exome and genome cases. And by the way, feel free to stop me at any point and ask questions. Through the ClinGen program, we've now operationalized this approach into these different categories of strength of evidence and developed terms for them: definitive, strong, moderate, limited, no evidence reported, at least in humans, or conflicting evidence that might lead you to either dispute the claim or in fact fully refute the original claim in the literature. And this framework is on our website. Now, the goal of this sort of systematic method that we've developed to evaluate the clinical validity of gene-disease pairs defines the criteria needed to assess that gene-disease validity, describes the strength of evidence supporting the gene-disease association in a semi-quantitative manner, and you'll see what I mean by semi-quantitative. It does allow users to methodically classify the validity using data that's not necessarily statistically computable, but it is not intended to replace valid statistical approaches that we hope all researchers, when they publish their new gene discoveries, will use. But it's really a way to take all of the variable types of data that we find when we look at gene disease causality and try to bring them together to give an overall sort of semi-quantitative assessment. So there's a work group that was formed through ClinGen to oversee this effort, and this gives you just a high-level view of what we're trying to do. Basically, we've developed this point scoring system to evaluate genetic evidence and experimental evidence. And so you get between 0 and 12 points towards the genetic evidence, 0 to 6 points towards experimental evidence, to give you up to 18 points. We then, in order to get from strong to definitive, it really takes replication and the test of time to make sure no refuting data has been published after that initial publication. So if it's been 3 years and there's at least 2 independent publications, then we can move it from the strong to the definitive category. We also track contradictory evidence, and that usually has to be not scored, but in fact evaluated to see if it refutes any of the basic data that's been published. And then we've set it up in terms of a workflow. So we have curators that work for ClinGen, some in my lab, some in other labs, who go through the literature and pull out all this evidence and assign points for a preliminary classification. And then we have experts in those disease areas come in and review all of that evidence and then either agree or they may decide to disagree and that some evidence wasn't appropriately considered based on their expertise in the biological systems and the phenotypes, et cetera, and they may slightly modify that. Oops, circles on these. So this gives you a little bit more detail in terms of that approach. So this is really scoring the genetic evidence. So that's things like case level data. And so if you have a condition that's dominant, then you would score the variance. And what you see here is a default point score for each case with a variant, each independent variant that you would assign. Now, that could be a proband that has a non-loss of function variant, but has some evidence for impact. Maybe there's some functional assay that the authors did to show that that variant has an impact on function. If you don't trust the assay very much, you might give it a very, 0.1 or something. If it's a very good functional assay, maybe you give it 1.5. It's sort of the range of points you're going to assign for each case that you've observed. If you observe a proband with a loss of function variant that's met the various criteria we talked about before, that gets a 1.5 points. Again, it can range depending on how convinced you are that that loss of function variant actually disrupts the protein. If the variant has been observed de novo, that gets a couple points. Again, it can range depending on the background rate of de novo variation in that gene. So you'd give Titin a very low score, but you might give a very small conserved gene a higher score for a given individual de novo variant. That's for dominant disease. For recessive disease, you get a certain number of points when you see 2 non-LOF variants in trans, or 2 variants in trans where there's at least 1 loss of function or de novo on one allele and then something else on the other. We also independently score segregation evidence based on the LOD score. And the higher the LOD score, the higher the number of points. So that's what's typically done for most very rare diseases. There are other genes that, and this really isn't meant to be yet applied to GWAS type associations, I should clarify, which is much better done with a pure statistical framework. However, we do hit genes where they're not, the monogenic phenotype that they're involved in is much less penetrant, and therefore a lot of those studies really are case control studies, and that they could be taking a single variant and doing a statistical analysis for its presence in cases versus control, or an aggregate variant analysis, kind of a burden effect, where you're looking at, if I sequence a group of patients with a particular disorder and you look at the burden of distinct mutations in affected individuals compared to the burden of mutations in unaffected. That could be burden of loss of function mutations or burden of missense variants, et cetera. So different types of aggregate analyses. And then, so each case could be given points for either the variant, or for both the variant and evidence, the independent evidence for each variant in the context of the appropriate mode of inheritance I just mentioned, as well as segregation data. So we went through this actually already in detail, same with the segregation evidence. So you can take a single LOD score and estimate across multiple families. So sometimes you have small pedigrees, like I showed you with that VCL, where you have 2 affected individuals in a family here, and 5 over here, and 3 here. So LOD scores are designed to be able to be additive. Although we do express caution if you start having lots of very small pedigrees, because there is the potential that the only ones out there that are published are the ones that positively segregated, and the ones that didn't segregate weren't published. So you have to be a little cautionary when you're trying to add up a bunch of really small families, and we have some sort of guidance against doing that to a large extent. We do give our curators, so a lot of papers don't actually publish LOD scores. And so if they do, you use the LOD score that was calculated by the authors, but sometimes the authors don't publish LOD scores, and you're left sort of adding up LOD scores across maybe multiple papers or for the paper itself that they didn't calculate it. So we give guidance to our curators on how to count segregations. Start with the proband, don't count them, 1, 2, 3, 4, etc. The approach to counting recessives is slightly different because you have to take into account the 2 alleles that are segregating, and you give a different point score for an affected versus the unaffected segregating. And then, as I mentioned, we have the case control data looking at single variant analysis versus the aggregate variant analysis, which each of these has to take into account the quality of the study design when you're looking at these case control studies. So these are some of the factors that we take into account for any given assay design. So the variant detection methodology that you're using for your cases versus your controls needs to be the same. You can't say, well, I did genotyping, in a, let's say, a burden effect. I used a SNP array for one population and then I sequenced the other. Not very good. Even different sequencing methodologies can sometimes give you inadequate variant detection in your 2 populations. So really wanting to make sure the methods you're using are the same for your cases and controls. Of course, the power, the number of cases, that's obviously important to assess the effect size. Any bias, in terms of the cases, their demographics, where are they from, how well were they phenotyped, making sure that your control population is adequately phenotyped for the absence of the phenotype. Now that usually, if they haven't been adequate at phenotype, is going to reduce your power, but nonetheless, it's important to have matching case control information for demographics. If you're doing your cases in a much more diverse population, perhaps an African population with a lot of variation, and your controls are not from Africans, you could see a very obvious burden effect difference by virtue of the genetic diversity in those 2 populations. And then, of course, basic statistical significance in terms of odds ratios, p-values must be taken into account. So that's sort of how we approach the case level evidence and scoring points for those different factors. On the experimental evidence, actually a group of us, Daniel, myself, and many others gathered a number of years ago to come up with guidance to really implicate genes in rare disease from a research perspective, and we took a lot of the functional approaches out of that paper that we published in Nature in 2014 to underlie our approach to scoring functional evidence. And the categories of functional evidence are in the area of biochemical function. So if the function of gene product B that you're asking for is very similar to A, and A is part of the same disease mechanism, that can help implicate gene product B, or the function of this particular gene product matches the actual phenotype in the patients. Maybe it's an enzyme with a particular function, and you can see buildup of a byproduct of that enzyme in the patient. So something about the patient's phenotype kind of specifically matches the gene function. Perhaps you've been able to document interaction, yeast hybrid assays, or other things that show that the proteins physically interact, and if one is implicated in that disease, it helps implicate the other. Expression studies, is the protein actually expressed in the tissue that the disease affects? Is expression actually altered in a patient with that disease through immunohistochemistry, other things? And then experimental evidence that shows functional alteration. So if you can demonstrate that the function of this protein is altered in patients with the disease, as well as through model systems, a knockout mouse model, for example, as well as rescue in a model where the gene function is knocked out. Can you replace it and restore function back to the model? So all those different types of biological or experimental assays then get scored. The biochemical protein interaction expression gets just a half a point each. Doesn't, causally implicate the gene, but you can gradually build up arguments. The functional alteration in patient cells gives you a little more points, and then the animal models and rescue models give the highest number of points. And then you can get anywhere, actually, I think an error said max was 6 points, it's actually to 8 points for the functional evidence. So then I mentioned that sometimes you find contradictory evidence in the literature. One report says one thing and another report says another. And how do you balance that? It's very difficult to score that sort of thing. It really requires an expert to come in and evaluate the contradictory evidence. And sometimes that contradictory evidence entirely refutes the claim. So I've seen papers where somebody, took their favorite gene and sequenced a bunch of patients, found, 5 missense variants, published it as a disease-causing mutation, and then somebody comes along like Daniel and publishes Exact, and every one of those variants is in like 20%, 30%, 50%, 80% of some, non-Caucasian population. I've seen huge numbers of genes just simply refuted solely on the allele frequency of all of the variants that were ever published in the original paper. So you could argue that that just entirely, and we have had the debate of, well, does that mean that you now go back to no evidence? Because essentially every bit of evidence in the original paper is entirely excluded. Or is that, disputed, or is that refuted? It's actually very hard to fully refute a gene's role in disease. You can say that there is no valid evidence, but to prove that that gene will never be implicated ever as a disease-causal gene actually is very tricky and hard to do. So, and we had this big debate in our committee. Well, if you've entirely refuted all the individual evidence, should you go back to no evidence category? And we ended up deciding to say that we would just use no evidence to mean no one's published any evidence in humans for clarity. And so when we find that all of the published evidence is essentially disputed, we put that into contradictory evidence, even though you can argue it should be no, but that's how we've sort of tried to develop some consistency in approach and clarify that at least somebody has made a claim. So anyway, long story short, we generally have to manually review and use experts to deal with the contradictory evidence to decide how to actually eventually classify it. So now I'm going to just go through some examples of actually using this scoring system for a gene. So this is the CD3E gene that's been implicated in SCID. So this is a disorder that leads to frequent recurrent infections beginning in infancy or childhood. It is an autosomal recessive disorder, and it's largely due to the absence of CD3 positive T cells. It has variable severity. It's T cell negative, B cell positive, and natural NK cell positive in terms of what you can observe in patients. So these were a series of 7 papers that our curators evaluated, documenting either functional evidence or clinical or case level evidence. And so largely they'll go through each of the references, they'll describe the cases and their phenotypes. It's actually very important to capture the phenotypes because they may have a very convincing mutation, but if the phenotype doesn't actually match the disease, you really can't count that as good evidence. That often is where our experts come in when they go and review the data the curators have brought in, and they'll say, gee, I'm looking at this phenotype. I actually don't think that's consistent with what I know is SCID. I mean, I'm not making a claim here, but that's what they'll come in and say, I know you've counted that, but I think that should not be counted because I don't think the phenotype matches. So that's the kind of thing our expert reviewers will do when they come in and look at this evidence. Was there a question? No. So here you can see there were 3 cases. I think these all do have convincing evidence of the disease. There were 2 biallelic heterozygous loss of function variants in trans, 2 homozygous cases. You can see the data in Exact shows that the variant is either absent or extremely rare. And of course, for a recessive disease, you often do see these variants present in Exact as long as it's below the frequency we would expect for carrier status, then you're okay. This one actually had RNA studies showing the complete loss of exon 2 in this particular case. They looked at segregation. There was really no segregation done in any of these families, but they were able to count 2 points for each of these 3 cases based on our framework. So that gave a final case level score of 6. They then looked at functional evidence. There's nothing biochemical that had been published. There was some protein interaction studies where this particular protein physically interacts with CD3G, which also leads to SCID when it's mutated. That gave them a half a point. The expression of the gene data was present in another paper looking at mRNA levels in patient samples. And then the protein was not expressed in a particular cell line. They got a couple points for that information. There is a gene disruption study looking at absence of this protein and its role in T cell development. Also an animal model that shows the effect of this particular protein. No rescue study. So that gave them 4 points towards function. So then they put all those are the 6 case level points, the 5.5 of the functional evidence, and that brings you to a total of 11.5, which is the total score here. There has been multiple publications over a span of more than 3 years for this particular gene. And so the curator classified it as strong. Now it's kind of like right on the border here. You round 11.5, you hit 12, and that's sort of when we would perhaps ask our experts to weigh in and say, do you believe this is strong evidence? And so, particularly, there are some genes where they're just incredibly rare causes of disease, and it is very hard to accumulate enough cases to get you to a high enough score. And so that's often when we sort of bring in our experts and say, you might not have quite got enough points here, but if you look at all of this data and what you know about SCID and the functional assays, do you think this is strong evidence? And so, in fact, oops, I didn't include it, but the expert did conclude that this was strong evidence and scored as such. So here's another example. This is a trickier one. This is PALB2. This has been implicated in hereditary breast cancer. So unlike SCID, this is not a very rare orphan disease. This is a much more common disorder. And when you search, you find lots of publications. So in this case, for case level evidence, they scored 6 different papers. Now there's both case control studies doing statistical comparisons and cases and controls. There's also individual cases in families. And so they ended up scoring, and some of these papers published both case control studies as well as individual case level data. And so they went through these cases controls that had odds ratios and were able to score. And this I have to say, this is the harder thing for some of our curators to do, is to really evaluate the power of these studies and how well they've been designed. And we often end up bringing experts into that process. But with these 3 different studies, these 2 looked at a specific mutation, looking at its presence in cases versus controls. This study looked at 2 different mutations. It could be scored as either a single or aggregate sort of analysis. It was scored really as 2 independent single studies in that case, or single variant studies. And then they looked, some of these papers looked at individual cases and scored, a small number of points for particular observation of a loss of function mutation in these 2 studies in individual patients. So, and there was some segregations from a couple of families as well. So in the end, they generated 10.9 points for case level data and 9 points for case control studies for this particular gene disease relationship. In addition, I didn't show you the functional evidence, but that was scored with 5 points. So in the end, that gave a sum of 17 points. And there have been multiple publications over many years that really then brought this to a definitive level, which was agreed by experts. So that gives you a sense of sort of how we go about the scoring of these, of the data, which involves lots of different pieces of information. It's very difficult to have a single statistical, assessment, although we hope each of the studies do valid statistical assessments. We also, I've been working, because Daniel and I have a grant through the Center for Mendelian Genomics. We've been trying to work to apply this method to the genes that are being discovered through the CMGs. So in a pilot study, we took 6 different genes that have been published from those groups and scored them. So this, I'm not going to go through extensively all of these assessments, but here's an example where there's a knockout mouse model, but it ended up scoring 0 points. Why? Because the actual specific disease that it's being looked at is Noonan syndrome, which is a dominant gain-of-function disease. So a knockout mouse model is not going to provide any evidence to actually implicate this particular gene-disease relationship. So that's, you know, example where you have to look at your model and say, is this actually a useful model or not? In this case, they scored it as 0, as not relevant to this particular assessment. And this, in this case, this particular gene was scored as moderate evidence based primarily on case level data with very little functional data to implicate this gene. And some, you know, there's a lot of genes where there's just 0 functional data out there at all. And so we often have to rely solely on case level observations. This is PSD3 and antecubital Brigham syndrome. There were only, basically only one proband with score, like this is an example of a single large family that's been published with no other data. And that, you know, there's 0.5 points for that one variant that seems reasonably impactful. And then there was a LOD score of 1.8. That's it for this entire gene-disease relationship. That gave 4.5 points in this, which brings you to limited evidence. So when that first paper comes out with one mutation, you know, could very possible, even though actually the LOD score of 1.8 is really not sufficient to implicate a first gene, and it could easily also have another variant in a nearby gene in linkage disequilibrium that is really the causal variant when you have only one mutation, particularly not a loss of function mutation. So this is an example. It's just simply limited at this point. So we've been starting to, we actually just submitted the paper to Nature Genetics on the methodology. We did a pilot assessment of maybe about 40 different genes, dual review with 2 different curators to compare the consistency in curation between different curators. And then when the expert review, how often did the expert agree with our curators versus change? So I actually didn't put that data up here, but that's all part of the paper that was just submitted 2 days ago to Nature Genetics. So hopefully that publication will come out soon. But we are already starting to put the curations that have been done by ClinGen on our website. So you can go and click on the ClinGen curated genes and diseases, and you'll get a screen that looks like this with all the genes that have some degree of curation done. And it could be actually in 4 different categories. We've had a long-standing effort in our cytogenetics community doing dosage sensitivity, scoring genes for haploinsufficiency and triplet sensitivity. So you'll see a lot of these in here. We have a group that does actionability assessment for a given gene disease pair and a specific intervention. It scores on the severity of the disease, the likelihood of disease, the effectiveness of the intervention, and the nature of the intervention. Is it removing an organ or just doing a screening test? And that gives you actionability assessments, which are also available. But the ones that I'm most interested for this is our gene disease validity. And so you'll see for the genes that this has been done, you'll see here definitive and definitive. You can then click on that. You'll get to a screen here. This is the Paul B2 one that I just showed you. It's classified as definitive. You can click on the report document, which will then open up that scoring matrix that I just showed you, and you'll be able to see, you know, how the points were assigned from different publications. Some of these are only the primary curator has done this, but we're making them available to the community even before we've gotten the experts to weigh in. In other cases, the expert has also weighed in, and you'll see the 2 different classifications that are there, which are usually the same. So that gives you a sense of how we're going about evaluating gene-disease relationships. Now, it'd be really nice for all these genes that are limited if we can get them to go one way or the other. So how do we actually move limited genes up the ladder? And this CMG exercise was actually based on published genes. There's a whole lot more genes that are sitting in people's databases, computers, that just don't have enough evidence to ever even get published. So how do we take these genes and build evidence to get them published and to get them, you know, evaluated in more patients? So I'm just going to present a case that was actually done here at the Brigham a number of years ago. You may recognize Anthony Philippakis, who works here, was originally involved in this case back when he was a fellow. So this was a patient with distal arthrogryposis type 5. At the time, the cause of this disease was unknown, although the actual phenotype had been described to either be inherited dominantly or showed de novo occurrence. And there was no known gene. The phenotype is quite complex with a lot of different organ systems involved. I won't go through the details of it. But we did whole genome sequencing on the trio, and we're able to identify 2 de novo mutations in coding sequence. Now, we all have, on average, 1 de novo coding mutation in our genomes. So if I just sequence you and your parents, I will find these in all of you. It doesn't mean it's going to go cause some disease in you. So when we find, a couple of genes with a de novo variant, we can't say, oh, well, that's clearly the cause of distal arthrogryposis type 5, right? It's a great candidate because that is a reasonably rare occurrence. But when you're looking across the genome at genes you have no rationale to believe are implicating your disease, it is really not very good evidence when you have 1 case. And in fact, there was another gene that also had a mutation in it. We actually ruled out this gene because there was quite common loss-of-function mutations in the general population, therefore unlikely to underlie this particular disease. And I should clarify, although observing de novo events across the genome in genes you haven't implicated in the disease, when you have implicated that gene in the disease, in the patient you're testing, it actually is pretty good evidence towards pathogenicity because you've now changed the prior probability of finding a de novo event when you narrow it down to just 1 or a few genes. But when you haven't associated those genes to your patient's disease yet, then that is a VUS, nothing else. So now in this particular case, we had this 1 case, no other data, which was not publishable. So the only approach that we thought was, well, maybe we can at least functionally prove that the variant disrupts the function of the protein, although I would argue that's still not useful. But when we reached out to this 1 guy in the world who studies this particular protein, he said, a week later, he said, oh, I got another call from another group in Canada, and they have a family with a different de novo mutation in the same gene, the exact same distal arthrogryposis type 5, incredibly rare phenotype. That is the evidence we needed to really causally implicate this particular gene in GA5. So piezo2 is the gene. It's now been replicated in many other studies. But the real evidence that came there was, in fact, the 2nd case, right? So we've now launched a program called the Matchmaker Exchange where you basically are trying to find patients with mutations somewhere around the world where you can match the phenotype and the individual gene candidate. So you're looking at overlapping phenotypic features between the 2 cases and at least 1 candidate gene overlapping between the 2 of them. Now this only really works if you're dealing with rare disease. Dealing with common diseases, matching 1 patient to another is not an effective statistical approach. And the Matchmaker Exchange builds off of a fundamental model of data sharing that's getting much more traction today in order to do global data sharing, which is a federated network, as opposed to a database where you try to convince everybody to put all their data in 1 place, which is very difficult, particularly internationally. The Matchmaker Exchange is what we call a federated network, where everybody owns their own database, and they use an API to allow communication between those databases. So in 2015, we launched this platform using 3 databases to start with, Phenome Central in Canada, Decipher in the UK, and GeneMatcher in the US, working with the Global Alliance on a lot of standards for API, data formats, regulatory and ethics, security approaches for authentication to try to get this network to work internationally between systems. Published a series of 17 papers on this sort of topic of matchmaking that's in human mutation. We're now at this stage, this is also still up to date as of today. We have 7 different systems. We've actually incorporated Matchbox system from Daniel's group into the Matchmaker Exchange. So now the rare disease cases we're doing here at the Broad, those candidate genes are now going through the matching system. And in addition to the 3 I mentioned, these three up here, and the Broad. We also have MyGene2, which is a patient-initiated, so patients put their own candidate genes in, maybe from clinical testing, other things. That's run by UW. Patient Archives in Australia has now launched and integrated their system. We also have Monarch Initiative, which is actually a number of disease ontology and databases that includes model organism data. So your candidate gene maybe doesn't match another human, but it might match a mouse model. And this is just the data fields that go into matching. We're running out of time, so I'm going to go a little fast through this. So we do have a website that kind of guides you through using the process either as a clinician, a patient, or if you want to deposit your own data set as a researcher, or hook up your entire database to the network. And we work with groups to bring them on board. I'm going to skip this. It's just how the matching is done. Basically, we need to match on candidate gene, and that's really all that's necessary to bring a match together, but then we score it based on phenotypic similarity. And so that's actually done because all the phenotypes are provided as structured terms through human phenotype ontology, and all of the individual human phenotypic features then map up through an ontological branch, so that even if somebody said, well, I have an abnormality of the eye, and somebody else said, I have deeply set eyes, even though those terms aren't the same because they're on the same ontological branch, you can get matching happening, and the degree of distance between each term contributes to the actual scoring of the phenotype match. That way you can have the system rank matches based on phenotypic similarity. So that's how the system, a lot of the different matchmakers use phenotype data to determine the strength of a match, even if it's the same candidate gene. For model organisms data, so a lot of model organisms supply evidence to human disease gene mapping. Now, the Monarch Initiative, in order to make this work, had to map all of the mouse phenotype terms to human terms. We use short stature, they use decreased body length. You can see all these different kinds of terms needed to be mapped so those ontological branches could be used to figure out that what you're describing in human actually matches what you're describing in a mouse. MyGene2, as I mentioned, is a patient-centric system, allows patients to go in and make their family profiles and enter the candidate genes that have been given back to them from a clinical report, and that's being used now to discover genes through matching that patients have started. So that's mostly what I wanted to cover. I have a couple cases that are just fun cases, but maybe I'll stop here. We have 5 minutes left and just see if there are questions about implicating genes and scoring evidence for gene disease causality. Yes? Yes?
If you combine, this is great. The Matchmaker sounds wonderful. But if you combine data sets from different sources, and before you mentioned how important it is to validate the quality of methodology, how do you do here?
Can you repeat the question?
So the question she's asking is, when we use Matchmaker Exchange, the data that's being put into these databases can be from all different methodologies, and how do we ensure that the quality of the data is the same when we're trying to do these matches? So largely today, the matching is not based on any technical data. It's simply that someone has put in and said, here's my candidate gene, ABC32 is I've considered a candidate from my own sequencing of this case. Now, I can tell you one of the biggest challenges practically in Matchmaker Exchange is when you have a match and then you go and follow up and you realize it's not a good match. In fact, most matches are not good. Now, they can be not good for a variety of reasons. They could be not good because the basis for calling it a candidate was fairly weak. For example, I gave you that DA5 case where our basis for that candidate was one of two de novo mutations in a trio, right? Now, other people might put in a candidate because it was one of 100 genes with biallelic mutations in like missense mutations in a recessive family. And that's all they had. They just went through and filtered on how many genes have a rare variant in both alleles. and they come up with 100, and they go enter 100 candidates. Well, you know that 99 of them are wrong, right, at the start. So they could then match some other case, and usually what happens is the two people get on the phone and say, well, my case has blah, 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 and the other person says, well, my case does not have that. They've got developmental delay, and you've got some, multi-congenital anomaly case, and they go, oh, not a match. And then sometimes but one thing that's been happening is that this has been helping rule out candidate genes. So usually people don't put 100 candidates in, but people do put multiples. And sometimes those candidates, you thought it was a de novo, but you didn't do a very good job with your trio sequencing. And in fact, the variant wasn't apparent, and the technical issues, are one of the problems that may underlie why it was a bad candidate to begin with. So at the end of the day, it's not a perfect system, and a lot of our efforts in Matchmaker Exchange are dedicated to trying to improve the quality of the match. So for example, the scoring now takes into account how many candidates you put in. If you put in 10, it divides your score by 10. So we don't yet have really good ways to sort of score the technical validity of the data, but at the end of the day, it sort of, we were starting to put in, well, what type of mutation? What's the evidence? Is it de novo, biallelic? Is it just a missense? Is it a loss of function, et cetera, that can then contribute to the scoring, et cetera. Okay, maybe time for one last question before we end. All right, it's you.
How often do you update the ClinGen data? Because new papers come out, new evidence is brought.
Yeah, so at the end of the day, it's a big project, but it's not so big that we're able to tackle all the genes. And so what we're thinking about for the new evidence is actually creating a portal on the website where anyone in the world can go say, new paper is out, or I published a new paper, and I think this needs to be updated. So it'll be a combination of our expert panels re-reviewing on a periodic basis, but also asking the community to alert us when new evidence has come out that we can then score. So it'll be both those things. All right, well, thank you very much for your attention. Hope you got something out of it.